Hello, good morning, and welcome back to the channel. It's always beautiful to have you here. This morning, we're going to be talking about citrate test, which happens to be the last test in the IMVIC battery of tests. And remember that we've been discussing the IMVIC battery of tests, where we look at different tests that can be used to differentiate members of the family Enterobacteriaceae. So, Escherichia coli, Shigella, Salmonella, we want to differentiate one from the other because they are members of the same family. They have very close genetic constitution or composition, and so it makes it sometimes difficult to differentiate one from the other. Now, citrate test is the last of the tests that we'll be discussing, and it's a test that it checks the ability of a bacteria to use citrate as its glucose source. Now, this is important. Remember that I told you that many of these tests, what they're trying to identify is the presence of a particular enzyme in the bacteria, or the ability of the bacteria to ferment or make use of particular different types of sugars for their respiratory activities and other metabolic activities. Now, in the citrate test, you are testing for two things or three things perhaps. One, you're testing for the ability of the bacteria to use citrate as its glucose source, so the bacteria can break down citrate and use it as a source of energy. Second, you're testing for the presence of an enzyme. The enzyme is called citrate permase. Citrate permase is the name of the enzyme, and it is present in bacteria that are able to make use of citrate as their source of glucose. So this is important. Also, it's important to know that apart from using citrate, the bacteria also make use of ammonium diphosphate as their source of nitrogen. So as they're making use of citrate as their source of sugar, they're making use of ammonium diphosphate as their source of nitrogen, because remember, most organisms need to make use of carbon, nitrogen, and oxygen for their different metabolic processes. So it's a simple test where we get to use a particular, well-designed agar that we call the Simon citrate agar. Simon citrate agar is used to run this test, and when the Simon citrate agar is prepared according to manufacturer's instruction, and then the organism of interest, the bacteria of interest, is inoculated into the Simon citrate agar, and then is incubated at between 35 to 37 degrees centigrade for 24 to 48 hours. Now, apart from that agar, we also add an indicator substance. In this instance, we add we add an indicator substance called bromothymol. Bromothymol blue is the name of the indicator substance that we also add, and then we are ready to go. Now, we allow this setup to incubate in the incubator for about 24 to 48 hours, and then we view. When we see growth of the organism along with blue coloration, that is giving us a positive result. Growth of the organism along with blue coloration gives us a positive result. If it is green in coloration, that is a negative result. So blue with growth is a positive result for us, and then we are ready to, and that blue indicates that there has been alkaline pH. That is, the pH of that medium has become alkaline. And so this is important for us to know. This is a test that we used to run in the IMVIC battery of tests. It's an important test, helps us to differentiate our different organisms one for the other. It's important to know that when it comes to being citrate positive, our Klebsiella pneumoniae is citrate positive, Enterobacter species are citrate positive, and our Salmonella are citrate positive, while our Escherichia and Shigella are citrate negative. This is important to know. We also remember that this test has a few limitations, in as much as it has a lot of advantages. Few of its limitations is that you need to use a pure culture. A pure culture has to be used to run it, and sometimes interpretation can be weak. If you don't have correct color change, interpretation becomes weak. Thank you for listening to this. This is just a short discourse of our citrate test, where we use the Simon citrate agar, and we use it to test for the ability of a bacteria to make use of citrate as a source of sugar. And that bacteria that's going to make use of citrate must have the enzyme citrate permase, and it's this enzyme that would help it. And thank you for listening. Remember to like, share our content, and do have a beautiful day.